Hello and welcome to all my students in the 13th lecture of your oral histology syllabus. This is Dr. Siham Al-Araj. I hope my voice finds you in good terms. We will be talking this time about the salivary glands. Roughly, we will be talking about gland architecture and in details about the major and the minor salivary glands. So, the lecture outline would be general organization, functions of saliva, methods of its secretion. We start about the parotid, submandibular, sublingual, and end with the minor salivary glands, and finally, some clinical considerations, as usual. The salivary glands so the salivary gland is relating to the saliva, which is the watery product in your mouth. A gland is a secretory tissue. They are compound, tubuloacinar, merocrine, and exocrine glands. The crine, it means also relating to secretions. We will be detailing each of these terms individually. So, first, compound, it means it's more than one tubule entering the main duct. Tubuloacinar, relating to the morphology of these glands. The merocrine, it means a secretion of the cells is secreted only. And exocrine means it's secreted into an outer surface and not into the blood, for example, or to the inside of the body. So, to be more specific about the terms, as we have promised, the secretory units have a morphology of tubuloacinar or tubuloalveolar, sometimes, and you found in some books. Tubule, it means it's the elongated structure, morphology. The acinus, which is a berry-like. And the alveolus or the sacs, the air sacs in the lungs, for example. This is where the secretions are released, so this is the morphology of the secretory cells. The ducts is the system where the secretions are transported. Again, describing the shape of the duct ends or the secretory units, which is found either in sacs or in tubules. This is the tubuloalveolar morphology. It can be simple or compound. Compound, it describes the complexity of the duct branching. So it's not a simple branching, it's a compound branching. Next, they are described as merocrine glands. Mere, it means only or just the secretions. Crine, the secretions are secreted. So, the proteins are synthesized in the Golgi complex, and the secretory vesicles are released in the lumen, the inside of the gland is called the lumen, where the secretions are moving out. Merocrine secretions, the cell is intact, only the vesicles are secreted outside of the cell. Lastly, there are exocrine secretions, which means they excrete to the outer surface of the mucosa. Here would be the oral mucosa, which we have studied in the last lectures. The general architecture of these glands is divided in two main components: the secretory tissue, which is the parenchyma, and the supporting connective tissue. We know always that the connective tissue serves for nutrition and support. From the stroma of the capsule, or is capsulated, a connective tissue capsule, we have septa, which divides the parenchyma into major lobes. All of this is a lobe, and which is further divided into lobules, small segments. So each segment or a lobe contains secretory units composed of acini, which are the secretory units that we've talked about, positioned around a lumen. The space in the inside of the acini. These secretions are secreted in the acini, and they empty into three sized ducts, from the smallest intercalated to striated into the largest collecting ducts. Both the intercalated and the striated ducts are in between the lobules, while the collecting ducts are found outside the lobules. Interlobular. The connective tissue septa, as we said, they carry the blood and nerve supply. There are fat cells which increase by age. The salivary secretions are undergoing the influence of autonomic nervous system, both sympathetic and parasympathetic. Again, this is all one lobe. It's separated into separate lobules by connective tissue septa. You can also find cells which encircle the lobules, we will talk about later. And the duct system, which is the intercalated and striated ducts inside the lobules, and the excretory ducts, which is outside. Morphologically speaking, some textbooks describe salivary glands as a bunch of grapes. The secretory units are each part of the grape that you eat. And we have the smallest to the largest branches. According to the type of secretion of these end pieces, they might be either serous or watery from serum, mucus texture, or a mix of a serous and mucus. So serous, it means watery, protein-rich fluid and a source of amylase, which is the enzyme for starch, the carbohydrates. The mucus cells, they contain proteins and carbohydrates, so they are more viscous, mucin-rich, or a mixed, which we have mucus cells and serous cells surrounding them. Lastly, we have the myoepithelial cell type surrounding these cells. Roughly talking, this is a diagrammatic representation of the two types of the end pieces, or what we call the acini, the serous and the mucus. Roughly speaking, the serous cells of this type have cell junctions in between in both types, while the serous end pieces or the serous cells may have what is called intercellular canaliculi, small canals in between the cells. Also, we have membrane enfoldings, what's called a brush border, both intercellularly and on the basal part of the cell. This is called the basal part because here is the basal lamina that separates it from the connective tissue. And here is the luminal part. The mucus end piece, on the other side, we don't see these small canaliculi in between the cells. Sometimes, as we said, it's a mixed secretion. We would have a serous also end piece in the end of the mucus tube, which is called a serous demilune or half moon, a crescent shaped end piece. Here are the central mucus cells around the lumen, and we see the serous demilunes. Moving ahead to the functions of saliva, which is the watery substance produced in the oral cavity. The mucin, which is a carbohydrated protein or a glycosylated protein, which enters in the composition of numerous mucus covering the cells in contact with the oral cavity. So this mucin, it serves as a lubrication to facilitate mastication, swallowing, and speech. Also, it forms a film that protects the mucosa, where hence comes the name, and keeping it a moist lining, as we said. Thirdly, the enzymes in the saliva brings the food substance into solution, which facilitates the taste by taste buds on the tongue, because when it's a solution, it's easier to form the function. Fourthly, it prevents the bacterial aggregation, meaning that if we have decreased level of saliva, there is higher aggregation of bacteria. The lysozyme, which is a non-specific antibacterial enzyme that lyses the bacterial wall. So it helps in the immune function against bacteria. Also, we have minerals that act as a buffer, resisting the pH changes in the oral cavity by the number of positive and negative ions. Next is epidermal growth factor, which is produced and important for the wound healing in the oral mucosa. Mucosal immune system by the IgA production. Also other types of antigens. And lastly, the amylase, which is the main constituent or the primary enzyme in the saliva, the most abundant, and it breaks down the carbohydrates into sugars. Amylase, the amylum is the starch or the carbohydrate. So this enzyme, it digests the carbohydrates, and it is mainly formed in the parotid gland. The saliva-producing tissues of the oral mucosa are classified upon either the size, major or minor, secretion, serous, mucus, or mixed. The major or bigger parts of glands are the parotid. Parotid, it means on the side of the face, so it comes on the side of the face near the ear. The submandibular beneath the mandible, and the sublingual beneath the tongue. Also, we have minor salivary glands in the overall of the oral mucosa, except in the gingiva and the anterior 2/3 of the dorsum of the tongue, where we have absence of minor salivary glands in the connective tissue. In the submucosa, sorry. The salivary secretions, normally we have low level throughout the day and increased throughout the night. Large additions at the meal times, because we said help swallowing and mastication and taste. The average flow is 0.3 milliliter per minute. According to the sublingual and the minor glands, they have a spontaneous mode of salivary secretion, while the parotid and the submandibular glands, they are nerve mediated and they're not spontaneous all the time secretion. During anesthesia, dental anesthesia, we have almost cessation of secretion. So all of the anesthetics, they depress the secretory cells. The mode of secretion of saliva can be by exocytosis of granules upon signal reception, or spontaneously through vesicular mechanism. The vesicles are built up constitutively from the Golgi complex to the plasma membrane. And third is the transcytosis, wherein substances move from the interstitial tissue across the cells. So the saliva passes transcellularly and or paracellularly in between the cells in the lumen. The cells making up the serous or the mucus end pieces are different in the electron microscope. The serous, as we said, comes from serum, which is a protein-rich fluid, or mucus from mucin, which is a more viscous glycosylated substance. Starting with the parotid gland, it is the largest salivary gland. It has a defined capsule, and it is mainly serous acini. We may find some mucus cells occasionally, especially in the young ages or infants. As usual, the septa divide the gland into lobes and lobules, and they contain connective tissue, vessels, nerves, and ducts. The lumina of the acini are serous acini are very narrow. Remember that for a watery fluid, we don't need a white lumen. The cells also have granular appearance, where we have the granules near the luminal side of the cell. The nuclei are round in shape and prominent, and they are located in the basal third of the cell. For more details of the serous cells under the electron microscope, they have a wedge or pyramidal shaped cells around the lumen. The basal lamina separates it from the connective tissue. This is the basal lamina. And we have what is called intracellular canaliculi that are here in between these cells, and they join the lumen. Both of the canaliculi are lined by microvilli, which is enfoldings of the plasma membrane. It's like a brush border. Remember that we always have desmosomes, tight and gap junctions, because they are epithelial-like cells. So as we said, water passes in these cells transcellularly across the cells and between the cells paracellularly. The proteins are packaged in granules, which are released through exocytosis when required. As we said, it's not a spontaneous secretion. The vesicles, they become more electron dense as they move towards the luminal end of the cell, meaning darker under the electron microscope. The appearance varies with the activity variation of the cell, meaning the granular appearance, and the resting cells have numerous what is called zymogen granules. Zymogen is a term for inactive enzyme or inactive proteins which are inside the granules of these cells. As we said, sympathetic and parasympathetic innervation of these acini. The nerve endings beneath the basal lamina comes in direct contact with the cellular membrane, called hypolemmal, beneath, or they remain on the other side of the basal lamina, over the cells. The parasympathetic drives fluid formation or the saliva production, and the sympathetic nerve supply drives increasing of the output, which is made by the contractile cells we would learn about later. Both, they cause the contraction of the myoepithelial cells. Now moving to the duct system, we have the intercalated, which is compressed in between the lobules, striated, which have the thin elongated stria, or the main collecting ducts. The smallest duct is the intercalated. They lead from the serous acini to the striated larger ducts. They are compressed, as we see by the arrows in the picture, in between the acini. They are lined by low cuboidal epithelial cells, and they have prominent nuclei with a small cytoplasm. These cells have smooth surfaces from the luminal and the basal sides. We see desmosomes in between the cells. We have occasional granules because they have low activity. Limited also number of organelles. They may act, this is important, as stem cells of the salivary glands. Next, several acini drain into each duct. So in one lobule, we find many intercalated ducts. In the parotid glands, they are characterized by being long, narrow, and branching. Remember that the parotid gland secretes a serous, a watery composition which has no problem to be driven or transported inside narrow intercalated ducts. Now the lumen in these ducts are wider than the serous acini, of course, but they stay the narrowest type of transporting ducts. The striated ducts are longer in the parotid gland and they are more active than the intercalated ducts. They have hence a larger cytoplasm, a large spherical and centrally located nucleus, and these cells are highly polarized. This is a characterization of the striated ducts as they are more columnar in shape. Now here what's different in these ducts are the microvilli, which are found in the luminal surface, and the striations on the basal surface. Stria, S-T-R-A-I-A, or with E for the plural, means the longitudinal parallel lines here. These striations, they represent enfoldings of the cell membrane, which is not regular. There are vertically aligned mitochondria, which you find here in between these foldings. This indicates that these cells have high mitochondria and are involved in active transport. Mitochondria are responsible for energy production. The role of these ducts are modification of the saliva by electrolyte secreted from the cells, secretory cells, by reabsorption of Na and Cl, and secretion of potassium and bicarbonate ions. Surely we have desmosomes. So the reabsorption is against the gradient, against the concentration gradient. Thus, it needs energy from the mitochondria and the unfoldings of the plasma membrane. Thus, it's called a stria, striated duct, because it's characterized by these striations. It converts the isotonic or the slightly hypertonic fluid into a hypotonic solution, which has less sodium chloride ions. A small secretory granules are found on the luminal side. The epidermal growth factor and the protein called kallikrein are characterization of these cells. As we said, these are active cells with more organelles and more granules than the previous intercalated ducts. These ducts are less abundant in the parotid compared to the submandibular gland, and they lead to the next collecting duct, which we'll talk about later. The largest ducts are called the collecting, excretory, or main duct. They have a columnar, unstriated, unaligned columnar cells, which gives the appearance of a pseudo-stratification, like multiple layers, but they are one layer only. They may have a layer of basal cells. It may have a layer of basal cells in some regions. The main parotid duct has two layers, a mucosal layer and a connective tissue adventitia, or an outer layer. Near the termination, of course, these ducts would become stratified multiple layers, and leading finally to the stratified squamous epithelium of the mucosa that we studied earlier. Now, the myoepithelial cells. These are non-secretory cells. They are found between the basal lamina and the membranes of both the secretory cells in the acini and the smallest intercalated duct cells. These cells have dendrites and radiating processes. They are rather longer or more elongated around the intercalated ducts with shorter processes. This when we compare it with the cells around the acinus, which have a fusiform shape and are much longer processes. They are even called basket cells because they have a catching appearance around the acini cluster of cells. Finally, they contract in response of sympathetic and parasympathetic stimulation. The name of myoepithelial cells come from them having cytokeratin intermediate filaments, which means they are epithelial in origin, and at the same time they are a specialized cell by having a smooth muscle-like cell because they have the actin microfilaments helping in the contraction function. Under the electron microscope, they have a flat nucleus, few protein synthesizing cells. They are linked by desmosomes to the secretory cells and by hemidesmosomes to the other side of the basal lamina, as you see in the picture. The function of these cells are firstly accelerating the outflow of the saliva when they contract around the secretory acinus. Secondly, reducing the luminal volume. Thus, they would increase or contribute to the secretory pressure. They support the parenchyma. Also, they have a supportive role. Fourthly, they reduce the backflow permeation of the fluid. They help to overcome any peripheral resistance by their highly radiating ends, cytoplasmic ends. They help the secretory cells to expel their content when they contract around them, and also they have milking effect on the fluid to assist the passage through tight junctions. Also, they are said to have tumor suppressing properties, preventing the invasion of epithelial tumors in the salivary glands. The submandibular gland is the second largest after the parotid. It is a mixed type secretion with mainly or a predominance of a serous acini. It has a capsule also and a serous acini that are similar to the parotid. Here in the picture you see more serous acini compared to the pink mucus acini. Now the serous acini, we said they are similar to the parotid. Concerning the mucus cells, they appear paler than the serous cells. In the hematoxylin and eosine stain, the nuclei are compressed basally, as you can see. They are compressed to the side of the basal lamina. Serous demilunes are crescent-shaped serous cells which appear under the microscope. The mucus cells can stain with the Alcian blue and the Pass periodic acid Schiff stain, which have the magenta or the purple color. Staining the mucin, especially the mucin. Here you see the paler cluster of cells are the mucus cells with a predominance of the serous acini, and sometimes they appear as crescent shape around the mucus cells. We see also the striated ducts here. So the serous cells have been shown to align with the mucus cells around a common lumen, while because of conventional tissue preparation, this leads to be pushed, the serous cells to be pushed aside towards the basal side of the acinus, giving the look of a demilune. The distension of the mucus granules also, it pushes the nucleus basally. That's why it's more basally located compared to the serous cells. These cells have limited organelles except for the Golgi apparatus or the Golgi complex. In comparison to the serous cells, we have, because more carbohydrates which are linked to the proteins, the Golgi complex is responsible for the glycosylation of the proteins that are produced from the endoplasmic reticulum, leading to the formation of mucus or mucigen granules. The pale secretory granules are discharged by exocytosis. The duct system of the submandibular gland is very similar to the parotid gland, with few differences. The intercalated ducts are much shorter. We said the parotid glands have especially long intercalated ducts, while here we hardly can locate the intercalated ducts. The striated ducts here, on the other hand, are longer and are more obvious. And now we'll see why. Because the submandibular gland has a predominant hypotonic or serous secretion, in which the striated ducts help in this function. Last of the major glands is the sublingual gland. It is made up of two parts, the greater and the lesser, the major sublingual gland, in addition to numerous minor glands. Each of the glands has its own duct, of course, its own duct system, and it empties into the sublingual fold. The major part of the sublingual gland is a mixed serous mucus, but mainly it is mucus. So remember both the submandibular and the sublingual gland, they empty into the sublingual fold, but the submandibular is mainly serous, the sublingual is mainly mucus, and hereby we have a balance of the type of saliva. So histologically we have group of pale mucus cells and occasional serous acini which look darker. Demilunes are similar to the submandibular gland. On the other hand, we have a less developed duct system. The striated ducts are usually absent, which makes the saliva more sodium rich and more towards the hypertonicity. Smaller parts of the gland drain into smaller ducts usually, and the major gland has more larger ducts system. Why we have less developed duct system when the secretion is more mucus in consistency? It is more difficult to be transported through the duct system, especially the very small in size. Hence, the sublingual gland has the lesser development of the duct system because it is the predominantly mucus. This is a representation of the serous acini, mucus acini, and when we have a mixed, we see a serous demilune with intercellular canaliculi. You see the myoepithelial cells around the acinus unit, the intercalated duct also, which have smooth surfaces, the striated duct, which have microvilli on the outer surface, luminal surface, and striations on the basal surface. This is as a summary. Next finally we talk about the minor salivary glands. They are located everywhere in the oral mucosa, buccal, labial, palatal, palatoglossal, and the lingual. We said except in the gingiva and the anterior 2/3 of the dorsum of tongue. These minor salivary glands are primary mucus, except for the von Ebner's gland, which are serous glands. Remember the von Ebner's glands, which empty their secretions in the trenches around the circumvallate papillae, which is on the tongue. Finally, another summary of the serous and the mucus secretory cell. We see the spheric in the serous part, spherical nucleus in the basal third, while a flat one compressed to the basal membrane in the mucus side. Zymogen granules rich in protein for the serous, while mucogen granules which are rich in glycosylated proteins. We have more organelles in the serous because it is more active in protein synthesis. We have limited organelles in the other hand. Golgi apparatus is more developed in the Golgi and more numerous in the mucus cells because they have the function of protein glycosylation and vesicle formation. Don't forget the intercellular caniculi in the serous cells, which are not present in the others. The desmosomes and gap junctions and tight junctions are present in both types of cells. Finally, the clinical considerations which you are asked to read about and know about are the xerostomia. Xero means dry and stomia is the mouth, so it's a reduced secretion of saliva. It is more prominent in the old patients, older age. Some medications may cause reduced salivary flow, also radiotherapy of the cancer tumors, and Sjogren's syndrome might have a clinical symptom of xerostomia. Now we have said that the unstimulated salivary flow is 0.3 milliliter per minute. The saliva has a very high role in the maintenance of oral health, so a reduced salivary flow would result in candidal infections, caries, and also periodontal diseases caused by the increased bacterial aggregation and the reduced prominent functions of the saliva. Sialoliths. The prefix sialo, it means salivary. Liths means a stone or calcification. So it would be a blockage of the main duct, usually occurring in the submandibular gland, and it may be a stone or calculus. It may cause a swelling of the gland caused by blockage of the outflow of saliva. At the end of this lecture, special thanks are addressed to Professor Firas Lahat for his support and help. I would like you to read this quote about the goal of true education. Hope you have benefited and consumed the information in this lecture, and I wish you happy studying. We would vote for a live session of questions after the end of the last lecture, if you like, and I'll be glad to answer any question sent to me and make some revisions for you before the exam. Have a good day.